So now let's begin with a mutation lecture in agriculture. So what are mutations? Mutations are genetic alterations, right? With the course of evolution, in our body also mutation is there, right? Addition, deletion. So that's why we have evolved from those apes and all, right? Being calling it of wheat, there also mutation had taken place, and we are able to eat that edible wheat, because as you know that wheat belongs to grass family. If mutation is not there, or evolutionary course of changes, the gene deletion, gene addition, and domestication would have not take place, then what would have happened? We would not be able to eat that wheat, edible wheat, which is a staple crop of the world, right? So now, moving on to mutation, why mutation are necessary? Some are harmful also, like what? Cancer. We are beginning with different kinds. We'll take in short. One is silent mutation, alteration in the DNA sequence that do not affect the encoded protein sequence, right? They are called as silent mutation. There is alteration in the DNA sequence which is not affecting the protein sequence. We know that after protein gene expression, proteins are very important, right? What we are having DNA, mRNA, then protein, and then expression, right? All these function are process proteins, right? So if protein is not getting altered, means we are safe. The character will get displayed, right? And now when we are moving, now we'll move to missense mutation. When there is substitution of single nucleotide that result in the change in the amino acid sequence of protein, it is called as missense mutation. Okay. Now next is nonsense mutation. Okay, what happens in nonsense mutation? When a premature stop codon comes in the way, right, all the processes will get affected and the process will shut down. So introduction of premature stop codon leading to truncated or non-functional protein. When prematurely stop codon UAA UGA will come, the proteins formation will get stopped, right? So nonsense mutations are harmful, right? One is frame shift mutation, right? We have, when we are reading the book, right, everything is written in a proper way. You suppose you are reading a point and a sentence, and two three words or words are jingled up and they are deleted or new words are added. You will see that, will you able to read it properly? Will the sentence make the sense? No. So similarly, if the nucleotides are removed, they are added or deleted, the biochemical processes will be there and will be affected. So frame shift mutation are, they occur due to insertion or deletion of nucleotides, right? So similarly, mutations are there due to either due to deletion, loss of chromosome, duplication means one extra copy is there, inversion, reversal of DNA, translocation means rearrangement of DNA, right? These are the certain factors due to which mutation is occurring, right? There were some basic slots we were discussing. Now moving on. We are discussing now the methods of inducing mutations. What are the methods of inducing mutation? Right? Chemically, how we are inducing it? One are alkylating agent. Alkyl group you will add necessarily in the DNA. DNA is having proper function. It is having all that benzene ring, and it is having all the A, T, G, C, everything. And the chemicals are going on, the process is going, and you are deliberately adding methyl groups. You see how the process will get affected, how our evolutionary biology will get affected, and you see that EMS, ethyl methane sulfonate, and methyl methane sulfonate, they are some agents, right? Another is base analogs. What are base analogs? They are 5-bromouracil or 2-aminopurine, right? They are also affecting the DNA. Another intercalculating agents, they are acridine dyes or ethidium bromide, right? Physical mutations, ionizing radiation, there are X-rays, gamma rays, there are also mutation. Ultraviolet rays are causing again mutation, right? Proton neutrons are causing mutation, right? Viruses, insertional mutagenesis, they are causing, they are the transposable elements which were discovered by whom? Barbara and McClintock in maize, right? We have ACDC, DS system in maize, IAS elements are there. They are beyond your scope. You'll study in later classes, slowly as we move up in course of agriculture, right? In viruses, I forgot, it is retrotransposons are there. They're like they're transposons, but they're in the viruses, right? Now this is very important technique, CIB technique. right? What happens in this? It was discovered by JC Kasha and Arrow. They're able to detect the mutation. What they are doing, they are having attached X, female is there, right? XXY, and male gamete is there. They are mutating the male, है ना? They are mutating the male, and now they are letting to breed male and female. What would happen? In the due course of nature, we know that female are carrier, because egg, for formation of female, X will be transferred from female, but here it is attached X chromosome, so X will not be transferred, so male will give. So when the male will give X, and we know that male was mutated, so we will able to identify the kind of mutation present over there, right? So this is CIB technique. CIB technique is widely used for detection of mutation. And you know that how you are detect means using mutation in agriculture. We have one developed variety, right? We have one developed variety. We want to introduce new variation over there. What we will do? What we will do? We will deliberately mutate it. Or what will happen in mutation? What is the basic behind idea to mutate things? That is to have recombinations or crossover. Huh? What are the things that are leading to the recombinations and everything? Huh? You know that mutation, hybridization. That's why studying of this mutation depth is very important. So